that we have the background out of the way, we can get into the mechanism of translation. How is translation initiated? What is the synthesis of the polypeptide process? And then how is it terminated? So we'll do the mechanism of translation. Now, before we do, let's kind of reorient ourselves again. Let's just kind of back off, look at the big picture again. We started with the DNA, a coding unit. We got a message, a primary transcript. If we were in eukaryotic cells, this would have a cap added to it, plus a poly A tail, introns removed. So we've got this mature message. So pre-mRNA. This would be mature mRNA. And again, this is eukaryotic. Here we've got our promoter. So let's zoom into this. We've got our cap. We've got the tail. Now, remember that the first codon to be transcribed is not the start of translation. The start of translation, whether it's prokaryotic or eukaryotic, is going to start at AUG. And if you remember, what is this sequence called that's upstream of there? This is the 5' untranslated region, the 5' UTR. Translation is going to stop at a stop codon, UGA. So this is actually, the UGA is not translated either, and that is part of the 3' UTR. This region that will be translated is the open reading frame. So this is our starting point. Now let's look at the initiation of translation. Here again is just sort of a depiction of where we're looking at. So whether it's prokaryotic or eukaryotic, the initiation of translation begins with the initiation complex. So an initiation complex forms, and it's always going to form near or at the 5' end of the mature message. And regardless if it's eukaryotes or prokaryotes, the complex contains initiation factors, also called IFs. Now, initiation factors are proteins that are going to be involved in the initiation of translation. Just like we had transcription factors that are involved in the initiation of transcription in eukaryotes, we had origination factors involved in the start of replication. We have initiation factors that are involved in the initiation of translation. Another component of the complex is the small ribosomal subunit, and a special transfer RNA called the initiator transfer RNA. Now, where the complex forms varies in prokaryotes versus eukaryotes. So let's take a look at the prokaryotic initiation complex first. In prokaryotes, the complex assembles at the Shine-Dalgarno sequence. So it's a sequence in the message. So it's a messenger RNA sequence called the Shine-Dalgarno sequence. And that sequence is 5' AGG, AGG, U. This is a Shine-Dalgarno sequence. And it is near the 5' end of the transcript. and it is upstream of the start codon. So it's upstream of the first AUG. So here's a little depiction. In green is our message, looking in 5' to 3' direction. In yellow, here in the cartoon yellow, if we zoom into that, we see our AGG, the Scheindahl-Garno sequence. It is near the 5' cap and it is upstream of the first start codon, where the reading frame is going to begin. And what symbols is, this is the small ribosomal subunit. Here are those initiation factors. And notice there's more than one. 1, 2, 3 of them. They assemble in a sort of ordered way, but you're not responsible for knowing that. Just know that there are multiple proteins called initiation factors that assemble with this complex. And then we have this initiator transfer RNA. And notice that the initiator transfer RNA, look at the anticodon. Can you zoom in? So if I draw this out, like the more we're used to seeing the transfer RNA, the sequence here, this is a 3' end. The sequence going 3' to 5' is AUG. Now, what is that going to base pair with? In the message, well, there's no cap here because we're in prokaryotes. In the message, it's going to base pair with AUG. So this initiator transfer RNA recognizes the AUG, and attached to it in this initiator transfer RNA is methionine, but it's a special methionine with an additional functional group called a formal group on it. So we're showing F-methionine. So the initiator transfer RNA recognizes AUG and it's charged with or has attached to it a formal methionine amino acid. So this is the complex that forms. In eukaryotes, the sequence that's recognized not sequence, but the complex actually assembles at that 5' cap. It's that special modified guanine that's stuck onto the end of the 5' end in that sort of reverse 5' to 5' triphosphate bond. And that's where eukaryotic assembly begins. So here is a eukaryotic, here's a 5' cap, here's a small ribosomal subunit. Here are initiation factors. This little E stands for eukaryotic, as opposed to prokaryotic. So these are eukaryotic initiation factors. Again, there's a number of them. And there is a special initiator transfer RNA that has the anticodon UAC that recognizes or will complement with AUG. It is not a formal methionine attached to the initiator transfer RNA. It is just with a normal old run-of-the-mill methionine attached to it. So this is the location where the complexes assemble, but we have not started the actual translation process. So it's either at Scheindahl-Garno in prokaryotes or at the 5' cap in eukaryotes. The components are initiation factors, small ribosomal subunit, and the initiator transfer RNA. So once the complex forms, the complex scans, it begins to move the message through. It scans in a 5' to 3' direction the message for the first AUG. Now remember, part of the complex is this initiator transfer RNA, which is going to hydrogen bond with AUG. So the initiator transfer RNA base pairs with AUG, and it's going to physically stop this moving complex, right? So if you can imagine, I don't know if I can make this look like it's moving, but here's, oh, sorry. Here's our initiation complex. It's scanning the message in a 5' to 3' direction. But when it gets here, it's going to stop because that transfer RNA is going to bind to it. So the initiator transfer RNA base pairs with AUG. The complex stalls or stops. And when this happens, the initiation factors dissociate. The complex stalls, initiation factors dissociate, and the large ribosomal subunit binds. So when it gets here, there's our complex, the initiation factors leave, and then the large subunit is going to come in and bind to it. Now this is what's going to do the translation. This is now the complete ribosome. And if you remember, I'm just going to clear this off, that the ribosomes are comprised of a small and a large subunit. These have different sort of names or densities depending on prokaryotes versus eukaryotes, but you don't need to know the 30S, 60S. I don't care about any of that. There's just a small and a large subunit, and each of those are comprised of protein components. Comprised of protein, polypeptide sequence, and specialized ribosomal RNA. And I just want to show you a depiction of one of these subunits. This is a prokaryotic 30S ribosomal, a small subunit of ribosomal subunit of prokaryotes. And look at here what you see in color. So this green portion, yellow portion, red portion, I see some protein helices, yellow. All that's the polypeptide component. But look at all this stuff in purple. What is in purple there is all ribosomal RNA. And here you can see, if you kind of take out the ribosomal RNA and view it, you can see all sorts of stem loop structures. We have some individual non-paired up RNA, but it's a massive sort of, ribosomal RNA is no joke, right? There's a lot of ribosomal RNA as part of this. And that RNA is going to play a role in orienting or holding in place the message and in bringing into place the transfer RNAs when it's time for the process of translation. Again, here's just sort of a depiction. Here's our initiation complex. This is a eukaryotic initiation complex scanning for the first AUG. In eukaryotes, the first AUG is in this specialized sequence called Kozak sequence. It stops here. The initiation factors dissociate and we get the assembly of the complete ribosome. And now translation is ready to begin. So let's look at the elongation or the synthesis phase. So synthesis of the polypeptide. So the synthesis in translation. So this ribosome, try to diagram it out again. It looks like a muffin. In the ribosome, there are 3 transfer RNA binding sites. This is in both eukaryotes and prokaryotes. There's an E site, a P site, and an A site. Between the large and the small subunit, the messenger RNA is sort of sandwiched in here, so that the codons, which would be here, the codons of the message are in these transfer RNA binding sites. The A site is also called the acceptor site. And this is where the magic's going to happen, because this initiator transfer RNA starts here. So our methionine, this would be then AUG starts. I'm sorry, not in the A site. This is in the P site. Oh, excuse me, P site. That is the peptidyl site, and that's where the process is going to begin. Now, in this A site, this is the acceptor site. This is where an incoming charged, remember that means a transfer RNA that is carrying its amino acid, an incoming charged transfer RNA that matches its appropriate codon comes in and binds. So we're going to have our second transfer RNA come in here and say it's carrying an alanine. When these are both they're coordinated here, they're inside this large subunit. This is where that peptidyl bond is going to form. The formation of the bond is actually going to shift this complex, and everything is going to move one unit. So a peptide bond forms between the amino acids in the P site and the A site. Now that's a covalent bond. Remember the bond? We formed a peptide bond in the last lecture. It's a covalent bond, so it doesn't occur by magic. It's catalyzed. And so this is actually catalyzed by peptidyl transferase activity in the large subunit of the ribosome. And so that's going to form the bond. And when that forms, this whole thing is going to move. Now, this initiator transfer RNA is going to let go of the methionine, and it's going to be shifted to the E site. So the transfer RNA in P loses its amino acid to the growing peptide. And it shifts into the E site. The E site is the exit, and it's released. Now this is hard because it's a moving process, so it's really hard to kind of do this in words written down on a piece of paper. So we'll look at it in a figure, and then we'll look at it in an animation. So this process is going to continue. New amino acids are going to come into that A site. New bonds are going to form. It's going to shift. It's going to shift by one codon. And so this process is repeated with a shift in one codon in a 5' to 3' direction each round. And it's repeated until termination. So when does termination occur? Termination of translation is simple enough. It occurs when a stop codon enters the A site. So when a termination codon enters the stop, when the stop codon enters the A site, there is no corresponding termination. Here we've got a stop codon in the A site. There's no corresponding transfer RNA. And since there is no transfer RNA, no bond forming, then it stalls the ribosome. So no corresponding transfer RNA. Ribosome stalls, and that allows for release factors to bind. So release factors bind, also called RS, bind. The ribosome disassembles or dissociates, falls off. Let's say disassembles. I'm not really sure if that's it. I think it is. Disassembles, and the peptide is released. So going back to the start, here we go with, this would be prokaryotic because it's forming the thymine in the P cell. There's the large subunit, the small subunit. Incoming charged transfer RNAs come into the A site. A peptide bond is formed by peptidotransferase activity. When this bond forms, it actually kind of pushes the complex or pushes the message the distance of one codon. And this will move into the E site and be released. And this transfer RNA with the growing peptide will be in the P site. The process continues and continues until termination. So let's watch an animation.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.